record ko lang po because this is our first meeting and I'll be discussing this for the first time with your section. So for module 7, it will be discussing cell division, specifically mitosis and meiosis, as well as the disorders which can affect us due to errors which can be found in cell division. Okay, so you've already studied about the phases, especially with mitosis, diba? So you've already discussed the phases with mitosis and meiosis, but I would just like to add a little bit more and discuss this a little bit. Okay, so for mitosis and meiosis, basically, it helps most organisms to alternate between generations. Okay, so generation from being a haploid and a diploid. In plants, their most notable alternation of generation is between the what we call the sporophyte and the gametophyte. Okay, so basically the sporophyte is the haploid generation. For us, the equivalent would be the gametes or the reproductive cells. This would be the sporophyte generation or the haploid generation. Okay, so the core concepts for cell division basically is that mitosis is usually used for somatic cells or body cells, and meiosis usually is for reproductive cells or the gametes. Okay, so DNA replication or the S phase, it is usually the process that is the first thing that needs to happen during cell division because it needs to create two copies of the DNA. Okay, so may ano lang, wala palang changes for DNA replication on both cell division. But basically, for the DNA replication, it has to happen for both cell division because doon sa mga magiging byproduct niya na cells, usually kailangan nun na copy ng DNA from the parent. So it needs to replicate this DNA para may pwede siyang ibigay dun sa magiging daughter cells niya. Okay? So napag-akalan na natin yung molecular signals, no? But sabi lang dito is that the gap phases, di ba there are gap phases or the gap phases or the G1 and the G2. So it's usually used to separate the S phase daw from the M phase or the mitosis or meiosis phase. Kasi this is the time when the signals, di ba, cyclin and no, not cyclin lang pala. This is usually the time when cyclin is either increasing or pagdating, di ba, ng around G0 or not G0, G1, di ba, time na nagbagsak siya or nawala siya. Okay, so usually yun yung, it's just there to mediate the molecular signals. Okay? So, ayan, alam na natin yan. Unregulated cell division can lead to cancer, but we'll discuss a little bit more later. Cell cycle checkpoints, like I said, it's there to check if tama ba yung mga nangyari doon sa buong phase natin sa cell division, like replication or kung, let's say, tama ba yung pag-divide ng chromosomes, pwede din, or nasa metaphase split ba sila or something like that. Okay? So mutations, mutations can be caused by a lot of things, diba? A lot of factors, but usually mutations in cell cycles, no, mutations in proteins involving the cell cycles or genes involving the cell cycle can lead to cancer or sorry, unregulated growth. Okay? So, pwedeng ganun or pwedeng magkaroon lang ng inefficiency or magkaroon ng damage dun sa mga certain genes na yun. Mamaya malalaman natin kung ano yung mga usual genes na involved when it comes to cancer. Okay? So, key roles for cell division, paulit-ulit tayo dito. For mitosis is usually for growth, development, for repair, and such. Okay? For meiosis, always, wala naman siyang ibang involvement bukod sa sexual reproduction. So for meiosis, it's always for um, reproduction as well as for diversity. Sa module nyo, makikita nyo that it has, yun nga, yung mga iba niyang functions. But basically, yun lang yung pinakaiba ng dalawa. Usually for somatic or for bodily processes, yung mitosis like repair and growth. And then for meiosis is usually for sexual reproduction. Okay? Okay, so some terms for the genetic material. Genome, basically, this means the collection of all the genes na nasa organism. So that's for the genome. Chromosomes, these are the condensed, diba? Condensed structures of um, DNA. So diba, this usually involves the coiling of the DNA to histones, blah, blah, blah. Okay? Chromatin, this is the what's chromatin again? This is the okay. This is the thin. So if chromosomes is the condensed version of the DNA, so chromatin would be uh DNA plus the histone, if I'm not mistaken. Okay, so diba I told you that the chromosomes is super coiled or kinocoil siya yung parang rope. For chromatin, this would be the earlier parts or the earlier stages of the coiling. Kasi it says here that it is the DNA molecules and associated proteins. The associated proteins would most probably be the histone. Okay? Sister chromatids, usually ito yung pair. Okay? Um, yung, di ba yung typical na nakikita natin na chromosome is yung parang butterfly siya. So yung dalawang pair of chromosome na yun, we usually term that as sister chromatids. Cohesins, so di ba, may sister chromatids ka. Cohesins would be the protein responsible for hindi attaching, for like binding both of these sister chromatids together. Okay, so those are cohesins. Sister chromatid cohesion is basically the process of kumbaga, sticking them together. Okay, centromere, okay, yung nasa gitna ng chromosome. So, di ba, it has shorter arms and then long arms. Yung nasa gitna, that is the centromere. Yung, ito, sandali pala pag tinuko ko. This one. Okay, so siya. Okay, so meron tayong isa pang part dyan, which is the centromere. Okay, so yan siya. Okay? Okay, so question. What if a chicken has 78 chromosomes in its somatic cells? Somatic or body cells. How many chromosomes did the chicken inherit from each parent? So how many? So if it has 78 chromosomes in its body cells, how many did the chicken inherit? Anyone? How many did it inherit? Or ilan yung namana niya from the mother, ilan yung namana niya from the father?
36.
Okay, so it's how many? 36 ba to? Ano ba dapat, ano na to? Okay, so it should be 39. Kasi 78 eh. So divide by 2. Tama, di ba? 39 yan. Hello?
Yes, sir.
Yes,
Okay,
sir.
so it should be 39. So 39 from mother, 39 from father. Okay, how about this one? How many chromosomes are in each of the chicken's gametes? Gametes meaning sex cells. Or kung babae siya, egg cell, chicken, yeah. So egg cell. How many?
39.
Okay, very good. So it should be 39. So since it has 78 chromosomes in its body or the somatic cells, mag-undergo ng meiosis, kailangan haploid yung sex cells niya, yung egg cells niya. So half of the original number. So if it's 78, then it should be half that, which is 39. How about for this one? How many chromosomes will be in each of the somatic cell of the chicken's offspring? So, ilang chromosome daw yung mapupunta sa mga body cells ng anak niya?
78.
Very good. Okay, it should be still 78. Okay, very good. So, gets nyo na siya. Okay, so let's move for the cell cycle. For the cell cycle, basically, ayan, M phase, di ba, we already know that this is the shortest phase, and the longest phase is the interphase, which involves the three subphases, which is G1, S phase, and G2. And again, there is such as a G0 stage, which is found in cells or which is observed in cells that are no longer actively dividing or no longer dividing at all. Okay? Ayan. So this is the M phase. It's relatively the shortest phase as compared to the rest of the cell cycle stages. Okay? So interphase mahaba siya. Okay. Discuss ko pa ba? So for ito, these are the highlights for G2. Basically, um, we still observe all of the things we can observe in a normal cell. Okay? Mm, this one. So this is the difference. Medyo different part lang is that the chromosomes are already duplicated but not condensed. Anyway, you can read more about that. Dito na ako pupunta sa mga phases talaga ng cell cycle natin. Okay? So let's start with the prophase. For the prophase, the main thing that you should remember for prophase is that you can already see the chromosomes with a microscope. Okay? So for prophase, it's always the condensing of the chromosome. Yung pag supercoil ng DNA, di ba? Usually the end product would be chromosome. And this chromosome is so dense, it is so coiled that you can already observe it using a light microscope. So imagine, you can already see the molecule. Hindi naman yung pinaka-structure of the DNA molecule, but rather, you have what we call, sobrang onti natin guys, kasi ko lang nakikita sa akin. Anyway, you have what we call, ano call this? Nakalimutan ko yung sasabihin ko. Okay, so basically basta sobrang nag-coil na yung DNA to the point that you can already see it under a microscope. Not the individual molecule, but rather yung collection niya. Okay, yung pagka-collecta niya or yung pagka-coil niya. Okay? Usually, yun lang yung talagang palatandaan natin dito. But you can also remember this part that sister chromatids joined at their centromeres, which is basically, di ba, yun nga, yung shorter arm then yung longer arm, di ba? So if you have here, ganyan, diba? These are the shorter arms, these are the longer arms. Sabi lang dito, the sister chromatids are joined at their centromeres. So basically, they're joined at that. Okay? Okay. Okay, so for meiosis, to be specific in meiosis part, dun sa prophase 1, which is yung first stage ng meiosis, diba? Meiosis is divided into two. For prophase 1, usually we indicate the number. For prophase 1, there are sub-stages pa. So if yeah. So for if sa cell division, 'di ba, we have the phases. Sa prophase 1, it is unique because it also has stages din. So it has sub-stages. Itong mga sub-stages or sub-phases na to are what we call the leptotene, zygote, pachytene, diplotin, and diakinesis. Okay? So usually hindi na siya dinidiscuss but I include it because it is very ano ba yung term? Basta kumbaga dito natin kasi makikita kung ano talagang nangyayari sa prophase 1. Okay? Kasi, for example, itong pachytene, the crossing over is very unique, di ba? Sorry. The crossing over is very unique to meiosis, di ba? Yung crossing over kasi yung nagpo-produce nung variability. Well, not just that, but it's part of the variation na nag-occur sa ating organisms, di ba? So crossing over is a unique part of the prophase 1 and it happens during the third stage or during the third sub-stage of prophase 1 which is the pachytene stage. Okay? So kung hihiwa-hiwalayin natin, basically leptotene is when chromosomes condense, zygote is when homologous chromosomes pair to form tetrads or bivalents. 'Di ba? Homologous chromosomes yung magkakasize sila. Okay, pachytene, pairing is completed and then nung na-complete na yung pairing, they undergo crossing over. So crossing over again, this is the part where they exchange, kumaga parang nagsaswap sila ng genetic material, 'di ba? And this produces variability or variation in organisms. Diplotin basically tatry mo na paghiwalayin yung homologous chromosomes but they remain attached to crossover points which we term as chiasma. So hindi buong chromosome yung nag um, nag nag-overlap sa each other during crossing over, but rather just certain parts of the chromosomes. Okay? Diakinesis, chromatids become visible. Okay? So basta, kumbaga, ang medyo highlight natin dito talaga is the packetid stage. But all of the other parts are also important. Okay? Yan. For metaphase, usually this is characterized by the migration of the chromosomes to the metaphase plate. So yung metaphase plate, basically, siya yung naghahati sa cell into two equal parts. Kumbaga, hinati mo siya sa kalahati. So, doon pipwesto yung chromosomes because, di ba, ipupull natin sila to divide them. Okay? So, yung pag-divide na yon usually, um, sa next stage pala yun, sa anaphase, di ba, yung pag-divide na yun. Pero usually, the metaphase is where these chromosomes should be placed dun sa gitna. Okay? Usually, yun yung palatandaan natin na naka-place na sila sa gitna or dun sa metaphase plate. Ay nakalagay yung equidistant para equal yung division natin dun sa cell. Okay? So, anaphase, this is considered as the shortest stage, and this is where the chromosomes um this is where the chromosomes are what do you call this, separated or divided or pulled. Okay? So sabi dito, please take note of this. Yung cohesin, di ba, it's the one that's attaching the sister chromatids together. It's cleaved or it is kumbaga pang cut. Okay? And sabi dito, each chromatid thus becomes an independent chromosome. So there is a stage within the cell cycle which doubles the number of the chromosome inside the cell. And this is during anaphase because kinakount yung chromatid as an independent chromosome. So for example, dito, for example, here, we started with it, how many is this? We started with 1, 2, 3, 4, 5, 6. 6 chromosomes. Okay, we had 6 chromosomes during this part. Okay, hindi ko natutuloy yung pagsusulat dyan. Chromosomes. Okay, 6 chromosomes. Then during anaphase rather, during anaphase, each individual here is counted as one chromosome. So if we had 6 originally, during anaphase we would have 12. Okay? But don't worry, during anaphase lang yan. Because for the next stage, usually, hindi na natin siya i-count as individual chromosome because nagko-close in na yung cell. Okay? Medyo nagko-close in na yung cell. So, we will consider these two as independent cells na. Okay? So, babalik ulit tayo dun sa number natin of original chromosome. So, here would have 6. Here would have 6. Okay? So, telophase and cytokinesis, basically, the cell is preparing for yung eventual talaga na pag-separate nila. Okay? Cytokinesis kasi is the term for the division of the cytoplasm. Meaning, talagang hiwalay na yung dalawang cell na yun. Wala na silang attachment point. Telophase basically is like the stage which prepares for the kumbaga, how do you term this? The telophase basically, it's like the preparation for the G1 phase ulit of the cell cycle. So, nucleoli reappear, kung last time wala yung nuclear envelope, slowly na siya nag-a-appear at nagpo-form sa telophase. So basically gusto niya na magbalik dun sa normal sa not normal, kumbaga dun sa non-divided stage niya. Okay? Okay. Okay, so what are the applications? So if you already read the module, cloning is one application um for these principles or the concepts that we discussed in mitosis and meiosis. So here we have Dolly, the sheep, which is the first cloned mammal. The first cloned mammal. Kasi after Dolly, it's usually followed by a lot more cloning methods, a lot more cloning applications. And if I'm not mistaken, the technology used for Dolly or the cloning procedure or cloning method used for Dolly is what we call the somatic cell nuclear transfer. And Dolly was from if I'm not mistaken again, a mammary gland, a somatic cell, tapos parang in-insertan na, basta parang ganun yung idea. You take out the cell, the somatic cell or the body cell, parang tatanggalin mo yung nucleus niya and then palitan mo ng um, I think a nucleus from a sex cell or from a gamete and then or a fertilized one. Basta parang basically the somatic cell would serve as like the surrogate. Parang ganun yung idea niya. I-house mo dun yung magiging future na organism. Okay? Here we have tissue culture. So, sa research last year, sa experimental research, tama ba? Madami pa na ganito. I think there was one group which engaged in tissue culture. So basically, you'll grow tissue. For example, plant. Pwede din animal tissue, but it usually involves a whole different aspect din. Pero for plants kasi, for plant tissues, you usually use what we call the agar. So pwede ka magpatubo ng plant tissues using agar. Okay, using agar. So, pwede ka magpatubo siya ng seedling or ng sprout and then i-transfer mo siya bago mo siya i-transfer. So, kung may treatment ka na gagawin, for example, you're treating it with like growth factors or and such, mabilis siya tutubo and then or you're trying to investigate a hormone or something like that. Pwede ganito yung setup niyo. Okay? So for disorders and diseases in mitosis and meiosis are involved in cell division. Sorry. We usually see or we usually observe what we call a karyotype. So I think you're familiar with this na nga dahil sa module, no? So it doesn't appear as like this one on the right, but usually it appears like this one. So you still have to pair this, or I think a computer software is used to pair the chromosomes together. How is it paired? It's usually paired using based on their sizes. Okay, so they have to be the same size kasi. So usually yeah, yun yeah. Usually they are paired based on their sizes. Okay? So here, for the karyotyping, we identify the number. And for humans, we have 23 chromosomes. Usually the diseases that we'll discuss here are what we call chromosomal aberrations or the ones having errors in the total number of the chromosomes. Okay? So we have. But first, one of the diseases and disorders that we need to tackle is cancer. Okay, so cancer, paulit-ulit na tayo sabi dito, this is the uncontrolled or unregulated cell division or yung pagdami ng cell natin. So usually that unregulated na pag-divide or the unregulated cell division is what produces the what we call tumors, diba? And what's amazing about cancer cells, hindi yung sa katawan natin kumbaga, what's amazing with cancer cells in a laboratory setting is that it usually does not involve kumbaga it can grow without external signals or without what we call growth factors. Kahit wala tayong growth factors, the cancer cell usually grows. That's why in most human cellular studies, ang model na ginagamit natin ay actually cancer cells. And this started with what we call the HeLa cells. Okay, so you can watch a video in TED-Ed. They made a video of um, intended about Henrietta Lacks, which is the person which they harvested the first HeLa cells. We term this as the immortal cell line. Parang yun yung term doon. Also, there's a book, I think, about the life of Henrietta Lacks. But, basta yun. So, basically, this started the fad with studying cell lines kasi yung HeLa cells talagang sobrang tibay niya. It can really grow. Kaya yung mga studies natin involving cells, usually yung nagamit nung una doon is the HeLa cells. Okay, but we have different lines na as of this moment, we have different cell lines na which we can use for studying different things. Okay? Next is yung sorry, what we call the metastasis. So the metastasis, this is the, sa Tagalog, we call this yung pagkalat ng cancer. So usually this is, parang we term this as the, ano ba yung tawag ko dun? Ba yun? Kumbaga transfer siguro or movement. Nakakatamad magsulat Transfer or movement nung cancer from one organ or from one place to another. So what causes metastasis? Sometimes it's in the nature of cancer. Sometimes during biopsies, if you're familiar with the biopsy is basically like you'll get a tissue sample. Okay? So usually, for example, internal, di ba? Let's say an organ is affected by cancer, let's say the stomach. So you want to get a biopsy to make sure what type of cancer that is or to identify what type of cancer that is. And sometimes during the procedure of a biopsy, may mga bits or like fragments of the tumor which is natatanggal tapos napupunta sa um, ating blood flow, sa ating blood vessel. So it will travel and pag nagsettle siya sa isang organ ulit, it can cause like another tumor to grow on that different organ. So sometimes, okay, sometimes biopsy can cause that, but not all the time. Okay, so there are also safe ways to diagnose cancer without the use of biopsies or with the use of biopsies. Iba-ibang method. Okay? So for cancer cells, also unique about them is that they promote angiogenesis. What's angiogenesis? Angiogenesis is basically the formation of blood vessels. So sir, bakit kaya niyang mag-promote ng angiogenesis? Ba't kaya niyang mag-produce or mag-promote ng pag-produce ng blood vessels? Because cancer cells are very um, voracious. Kumbaga gusto nila madami silang nutrients, madami silang food. So in order to do that, in order for it to grow more, for the tumor to grow more, it produces blood vessels or it promotes the formation of blood vessels para may magkaroon siya ng blood vessels, magkaroon siya ng constant supply of nutrients and um, oxygen. And usually, di ba, kaya napapansin natin for people with cancer, they really grow thin, they really grow, kumbaga, yun nga, umaano sila, pumapayat sila because naaagaw yung nutrients nila na pupunta sa tumor. Okay? Okay, so these are the genes that I was telling you about a while ago. Oncogenes are basically the genes that are has the possibility to cause unregulated cell growth. Okay? Oncogenes are the genes that has the capability to um, not produce. It has the capability to instruct the cell go, no, what am I saying? Wait lang. Oncogenes basically are the genes responsible for causing the formation of tumor or for causing unregulated cell division. Okay? Bakit may gene, sir, may genes po tayo na nagkakos ng cancer? Not specifically. But, for example, we have a gene which controls the, um, the signaling or the production of cyclin, for example. Kunyari, nag-mutate yon and during this mutation or during this yung error dun sa gene na yon, ano, um, na-overexpress siya or continuous siyang na-express ng katawan, paulit na ine-express. So kung yung cell na yon laging may cyclin, mataas yung concentration niya ng cyclin, we would expect that the a cell would keep undergoing cell division. So in that sense, the gene which controls the cyclin, the mutated gene which controls the production of cyclin, is an oncogene. Okay, kasi it caused the unregulated cell division. Okay, but there are a lot of genes which can do the damage or which can lead to cancer. Okay? Kaya nga yung term is oncogenes kasi madami sila. Onco, 'di ba, for cancer, 'di ba, oncology, blah blah blah. So oncogenes, these are the genes if mutated or which if are damaged can cause cancer. Okay? But again, our bodies actually have a way to suppress tumors or to counteract cancer in the form of the tumor suppressor gene. So normally, our bodies have these genes which give us this ability na, for example, as of this moment, kunyari ako, there's a cell na nagkakaroon ng error. It's constantly dividing. But I also have a gene which can counteract that, which is the tumor suppressor gene. So basically, it can negate cancer or it can negate tumor formation. So if ganun pala sir, dapat walang may cancer. But again, sometimes if genes produce if normal genes can become oncogenes, tumor suppressor genes can also be faulty or pwedeng maging ano siya, inefficient or faulty. So if it's inefficient or faulty, meaning it can't do its job. So magkakaroon pa rin ako no problem if faulty na yung gene na yun. Okay? So basically ganun po siya. So cancer, di ba, we have also a lot of types of cancer. Okay? So let's move to chromosomal aberration. Sorry. So let's move to chromosomal aberrations, another disorder or disease which involves the cell division. No? Most of these diseases, or actually most of the abnormality that we can observe in chromosomes is due to what we call non-disjunction. What is non-disjunction? Non-disjunction. Okay. What is non-disjunction? Non-disjunction is basically um, the failure of these chromosomes to divide properly. So, kunyari, hindi siya na-divide equally, like tatlo yung nasama instead of isa lang, or mali yung pairing sa meiosis. Those are all factors which can affect the separation of these chromosomes, di ba? But mostly these are due to errors nga nung paghiwalay. So sa anaphase natin nakikita kasi yung non-disjunction, ano? So non-disjunction would produce then an unequal or an abnormal number of chromosomes um in a certain number. For example, chromosome 21. So due to non-disjunction, it produces what we call a trisomy o yung tatlong kopya ng chromosome. So trisomy 21 is also known, di ba, as Down syndrome. Sorry, not Down syndrome. Okay, so that's an example of a chromosomal aberration. Here we also have trisomy 13 or pataw. I think this is pataw. You can search what this is. So basically, ganun din po siya. Trisomy 18, meron din. I think this is Edwards. Nakalimutan ko na. Basta, may disease din yan. If I'm not mistaken, Edwards. Ayan, this one is pretty good. So, minsan din, nangyayari, nade-delete or natatanggal yung chromosome. Okay? So, pag na-delete or nagkaroon ng deletion, it's another chromosomal aberration. So, one example is the deletion of the Q arm. Q arm to, if I'm not mistaken, the Q is for the short one. The Q arm or the short arm, yung deletion ng short arm na yon, it will cause what we call the Cri-du-Chat syndrome or Cri-du-Chat disease. So this is the deletion of the fifth chromosome. Okay, deletion of one arm of the fifth chromosome. This one for the deletion of 11, I think this is yung Jacobsen. I think this is for the deletion of chromosome 11 or one part ng chromosome 11. Doon sa P arm, may nabawasan siyang part, may natanggal siya na part or na-delete. So, pwede din sa sex chromosomes siya. So, if you're familiar with sex chromosomes, diba, ito yung X at Y. Usually, pwedeng may multiple copies ng X, wala si Y pala, yung mga ganon. So, dito, missing lang siya ng isang X. So, X0 siya. Okay, so I think this is Turner syndrome, if I'm not mistaken. Turner, Klinefelter, yung mga ganon. Okay? Okay, do you have any questions about our topic? Whew. Any questions? Okay, so tomorrow, hopefully, we finish the discussion on, wait,